Did you know that thanks to climate change, tacos could disappear? Corn is the main ingredient in Mexican nutrition. Currently, we consume 196 kilograms annually per capita. However, national production is not enough to satisfy the needs of the population. Tacos could disappear. Environmental, land, corn, and logistic factors is the main ingredient in Mexican nutrition. Is really sensitive to temperature changes. We consume 196 kilograms. The states that produce the most corn per capita are Guanajuato. However, national production is not enough to satisfy the needs of the population. Tacos could disappear. Environmental, land, not enough.
I think we had a problem. Give me a second.
Guanajuato, Sonora, Sinaloa, Jalisco, and the state of Mexico. All of them are severely affected by droughts and frosts, leading to losses of at least $88 million, representing around 90% of all crops in Sinaloa and other states. Thanks to synthetic biology, however, we can solve this problem using a yeast that lives in the corn root. This yeast is designed to produce an anti-freezing protein that will also help reduce dehydration in the plant, allowing it to survive in more extreme conditions. We will regulate the system with humidity and temperature sensors that will give a lactose solution which activates the yeast. In this way, the efficiency of crops can be increased in diverse environments, improving agriculture as well as the consumption of local corn, at some point in the near future taking it to a large scale. And we are BioWolves.
Can start with the questions now. If any judge wants to start, you can raise your hand. Kevin, please.
Hi, everyone. So I have a few questions. I will start with one right now. I want to know, because the system that you propose use glucose and lactose, right? So I want to ask, is there any potential effect or potential biohazard of manually adding glucose or lactose to the soil? Because you are planning to do that, if I understand.
Yeah, sure. Well, the main solution is going to be made of galactose, and well, the main effect is the change of pH on the soil. However, it is a change of pH that will favor the corn, since it prefers more acidic soils. The pH that the corn prefers is between 5.5 and 7, while the galactose has a pH around 6 and 6.5. On our parts, there is not a lot of organisms that metabolize galactose that you see. Yes, there are some that metabolize it. However, due to certain parts of the pathway, it ends up being toxic to them. For example, like certain species of fungi can't completely metabolize it unless there is another carbon sources. So really, there will not be any kind of biohazard, only the change of pH, and in any case, in kind of a pesticide action that will benefit us as well.
Okay, thank you.
Thank you. Harold, please.
Yes, hi everyone. I really like your project. My question is the following. You use a yeast as a host for your genetic circuit, what was the advantage of use a yeast instead of a bacteria like E. coli?
Well, just the main advantage, for example, in this case, is that yeast formed these symbiotic relationships with corn that allow them to pass directly proteins and other resources. Most of the yeast or rhizosphere yeast that form these symbiotic relationships with corn help them in growth promotion factors and protein delivery. So that's like the main advantage, because we already have like that system that will allow the plant to receive the protein without the plant being considered a GMO. So that would be like the main advantage of using a bacteria, as well as, for example, there are a lot of yeast populations present in the roots of the corn, so it would be like more easily to have like this connection with the agricultors without them having like a concern about the safety.
Thank you.
Michelle, please.
Hi, everyone. Congrats, guys, for your work. My question is, there is a scientific article where Guo and her group used data mining, and they found stress responses in plants are similar for cold, water, and soil stress. So it will be interesting for you to study how your project could help with the problem of soil stress. Since part of Mexico, for example, Sonora, Sinaloa, Nuevo León, Oaxaca, Veracruz, and other parts are affected by saline soils. Have you ever considered that?
So in this case, we consider to start building our idea upon how can this mycorrhizal interaction can help the soil, and for our purpose is to diminish the water use for crop production.
Thank you. Any other questions from the judges? Yes, Kevin, go ahead.
Yeah. Another question is that your system is designed to act basically against temperature. Yes. So do you consider like how can the system, be sensitive to temperature itself? Like, have you thought about some circuit of some upgrades that could allow your system to specifically react against temperature and not against glucose, galactose?
Yes, Hugo.
Yeah, sorry. It was considered at the beginning to use some promoters that are able to be activated when, for example, freezing is detected, or even for desiccation. The problem is that they are not well known, and moreover, the first problem that we have is that the mycorrhiza, it's on the dirt, it's on the soil. So the conditions that are in the dirt, because if you, for example, this was the first proposition, right? If you put your hand under dirt, the temperature is kind of different. It is warmer when it is freezing outside, or even it is cold when it is completely hot outside. So the idea is to, I mean, we already have like a prototype for design that works with Arduino, with NLME35 sensors and humidity sensors, in order to detect the conditions outside without the, I mean, it can't be itself, because it can detect the plant conditions. So we are able to modify it electronically and then deliver the galactose in order to activate the genetic circuit and make all that stuff. But yeah, it was already considered.
To complement what Hugo said, also, for example, we could maybe add a system where the current was the one responsible for detecting the temperature, but we then will lose our advantage of not having a GMO product. So that's the reason why we prefer to go with the digital systems to detect temperature.
Okay, thank you.
Thank you. Any more questions? Yes, Beda, go ahead. Oh, you're muted, I'm sorry.
First of all, congrats on this project, which I found very interesting and also quite ambitious. In order to make a sensible question, I need to understand first a little bit. I'm sure to be sure to understand. So you would apply that modified yeast beforehand to the soil, if I understood correctly, where it would somehow be part of a mycorrhiza system, and you would activate it with applying lactose or whatever to the soil shortly before you have a temperature problem, low temperatures. And then that yeast would produce an antifreeze compound, which would enter the plant, I understand, and go to the organs of the plant that might die due to cold temperatures. Is that more or less correct?
Yes, more or less. The only, well, to make a little change, in this case, we will have their yeast already has like a evolutionary relationship with the yeast, with the plant, sorry. So by introducing our modified yeast, it will form their symbiotic relationship with the straw.
Directly, the yeast with corn.
Yeah. It's a
Okay.
species called Gerosima guillermondi, and they already have proven to have a relationship. So by only introducing it, it will form the relationship without any problem. So that's our, that's
Okay.
just it.
So if that's more or less correct, my question would be, first of all, how does that yeast compete with other yeasts or other microbiological entities that live in the soil? What advantages does it have? And second question, how does the antifreeze go to the growth point, which is, according to the farmer you interviewed in one of the videos, it's the most sensitive point of the plant that might die in cold temperatures.
Excuse me, just to rectify. Oh, I think I have some echo. Could you repeat? The question is exactly how does it competes? I mean, just competing, and there's no other question. But okay, so answering to that, this mycorrhiza does not have any advantage over others. And actually it interacts with all the biosphere, and it's called another way of the roots. So even if we if this mycorrhiza is modified to produce the antifreezing protein, the relation it's already found in many other maize plants. So in that way, even if we are administrating galactose, it will compete in the same way as other microorganisms. And the galactose metabolism haven't been touched, so it will process the galactose in the same ways as other organisms. So answering to that question, I mean, it's like we do actually don't know what's the competition of it has any other advantage, but as it is already in the environment and it's already found in maize plants, in mycorrhiza, we can say that, I mean, there's like no any other problem by just adding the antifreezing protein to the system. It is just the advantage is going to be, I mean, the only part that is benefit from this is the plant that is receiving the antifreezing protein. Other advantages are just that they are more resistant to the freeze temperature and even to desiccation. I don't know, because this is something that we have to clarify. and I was about to say that the antifreezing protein is not just protecting against the low temperatures. It is protecting against the desiccation, as it avoids decrease the evaporation of the water. So that's the only advantage. But any other organisms that are in that environment, near to the roots where it's the mycorrhiza, they are going to have the same benefits because it is excreted.
Thank you. We'll go with Harold, please.
Yeah. In the process of modeling your genetic circuit, how the model help you to develop a better parameters or something to improve the mathematical model that you do?
Anyone from the team who would like to reply to the question? Yes, we'll go.
Yeah, sorry. From the model.
You're mute.
I was mute. Yeah, sorry. From the model, it was the things that we obtain is that we have like a redundant circuit that will lower a little bit the secretion, the expression of the antifreezing protein, and that's something that helps us to know that there are some interactions between the GAL promoter and the galactose, but it is through a specific protein that is considered in the circuit, which actually it is MIG1, and that's why it was proposed to repress that gene from the yeast in order to have an improvement in the secretion, in the initial reception of the galactose, and that improve that sensitive. And even from the model, we see that it was necessary to overexpress GAL3 and GAL4 proteins in order to improve even more the sensitivity of the PIWIL1 promoter. And actually, it from the mathematical models, it was just used from previous articles, because that's something that actually we lacked a little, and it's to put all that in a single system, as it is not known at all. For example, the behavior of the promoter IMA1 that is inside the cell. The PIWIL1, there is still be some misconceptions in other articles where it is not well known which other proteins are interacting with PIWIL1 promoter. So something that we can improve completely is maybe by having more information from other articles or in making it more practically, because theoretically it is a little hard to develop a well mathematical model.
Thank you. Kevin?
Yes. So how big is exactly this antifreeze component, and how is the antifreeze component exported to the roots of the plants,
Okay, so this protein is considerably smaller, for example, of GFP. We are basing this mechanism in an article that was made by understanding the way plants absorb proteins through their roots. And they observed that, for example, GFP, that is a very big protein, could pass without problem. And they would detect in different tissues, including root tissues and stem cell tissues. And for example, considering that GFP is a very big protein, we consider that ours, that it's considerably smaller, wouldn't have any kind of problem to pass. The mechanism where the protein is transported across the plant is not quite understood. So we don't know exactly how it's done. We only know that yes, our plant will pass without problem. The protein will pass without problem. But I will give the word to Hugo.
Yeah. Also, for the let's divide this excretion and reception, right? Because there are those are the two questions. First, from the excretion, it was used previously researched modified synthetic propeptide that is used in as Saccharomyces cerevisiae, and even it was researched in Pichia pastoris. And its way it works, it is from a toxin. I mean, it is not toxic, the propeptide, but the toxins are something that needs to be excreted, right? That amount of protein that was mechanism, and that's why some, for example, we also consider to use the yeast. It is more known that the mechanism of excretion, better known the mechanism of excretion, because actually that happens with other proteins. And this propeptide sequence, it's allowing through the endoplasmatic reticulum to go outside of the yeast. And then, as Diego already mentioned, we was a little concerned if the plant is going to be able to recept this 30 kilodaltons protein. As I know, a little bit less. It was 27 or 30 kilodaltons, the antifreezing proteins. And the GFP that it was compared with, it was 32 kilodaltons proteins. I mean, from other studies. So it was observed that the plant was able to absorb even that antifreezing protein through the roots, and maybe it's traveled through the plant. And in other articles, because it wasn't known that the proteins were able to travel through the plant. But then with this article proved that this through the xylem, it was able to transport the protein. But after some time, it was degraded. It was through using proteases, and that's why we were considering that maybe it can travel to the corn, but it was too at the top that it wasn't able to travel to the top of the plant. So we are not consuming the antifreezing protein. And I mean, there are many things that were considered, but yeah, the size is not that big compared to GFP, and the mechanism of action of the excretion is through a propeptide.
Also, we need to consider that since the relationship, the yeast is an endophytic, has an endophytic relationships, the absorption is not directly to the roots, so it's more like delivered directly to the plant. So it will also make more easily the transport of the protein.
Okay, thank you.
Thank you. Any more questions from the judges? We have 1 minute. Please just remember, advisors and mentors are not allowed to ask questions or reply to any questions. We have 1 minute till we finish this session. Yes, Beba, go ahead.
Another practical point. When you want to apply this galactose, when you have a drought, you will have a very dry soil. And if you want to apply that in a watery solution, I don't see how that gets to most of the roots unless you apply huge volumes of water. That probably stay on top of the soil. Might be quite just a minor problem you can solve somehow, but just occurred to me right now. Any thoughts?
So we will leave that as a feedback. So thank you. The session is finished for this team. So I will ask all of the team members from BioWolf to please step out of the room so we can move on to the next one. Thank you, everyone.
Thank you.
Thank you.
Thank you.
Kevin, so if you can please leave the session.
Me?
No, the student. I will.
Okay.
I will now allow to comment the next team. So we will start with IGN Biotech Easy. Welcome team members from IGN Biotech EC. If you still have more team members that want to join, please just let them know that they need to add your team name to their Zoom name so I can let them in. I will start sharing with your promotional video, and then you will have 20 minutes to answer questions from the judges.
¿En realidad sabes lo que comes? ¿Qué hay en tus alimentos cuando los compras más allá de lo que puedes ver? En la carne crecen muchos microorganismos que liberan sustancias tóxicas, como la putrescina, encargada de dar ese olor tan desagradable. Como ejemplo, llegas a casa, comes tu carne en familia y ¡oh no! La intoxicación alimentaria es más común de lo que crees. En América Latina se reportan 600 millones de casos y Ecuador no es una excepción a la regla. Pero ¿qué pasaría si pudiéramos cambiar el juego y prevenir estas enfermedades antes de que ocurran? Como investigadores, hemos propuesto la creación de Pop Fresh Plus, una etiqueta inteligente bicolor que te ayuda a saber si la carne puede consumirse o no. Cuando la etiqueta se encuentra de color azul, es posible comer. Pero cuando está rojo, es mejor que no lo hagas.
Para que se exprese el azul y rojo, se utiliza un sistema libre de células que trabaja como detector de putrescina. Sí, el que genera el mal olor en la carne. Y te avisa si es seguro consumir. Nuestro sistema libre de células está formado por componentes celulares, como ribosomas y polimerasas, junto a un circuito biológico que detecta la presencia de putrescina. En pequeñas cantidades se producirá una reacción azul, pero encima del límite se pintará de rojo, pudiendo ser aplicada a la detección de otros tipos de alimentos o incluso enfermedades.
En un mundo donde cada bocado importa, Pop Fresh Plus busca poner el poder de la salud en tus manos.
Do you have any questions from the judges? We have 20 minutes. We'll start with Kevin.
Hi everyone. First of all, great work. I have a question. It's about if you have ideas of what other kind of methods currently exist to measure putrescine, and how does your solution differ from those methods?
Okay, yes, there are other laboratory methods, techniques in order to sense or even measure putrescine. However, they tend to be expensive. In our case, we decided to include a quorum sense design, a biosensor that uses it is like a cell-free biosensor. And we decided to include a quorum sensing model in order to make all the components in a bioreactor without the need to add an inductive like IPTG or those things. So it is somehow cost effective. It's cheaper than other methods.
I would like to add something. There are other methods, but they don't specifically detect putrescine. For example, I don't know, in the MERS, there is the possibility to detect the pH, but the pH is not necessarily related with the putrefaction of the meat. So we decided to focus on a molecule that absolutely confirms that if the meat is degrading, you have to detect putrescine. So we difference on the sense that we are using biotechnology engineer and not chemical reactions, like, for example, detecting by the pH. That's one of our principal difference.
Okay, thank you.
Thank you. Harold?
Thank you. I really like the project and how you model the genetic circuit. My question is the following. In your project, you develop some mathematical equation and you got some different equation. What were the assumptions that you taking into account to develop this mathematical model?
Yes. In the mathematical model, we assume that the sources inside our self-free system is limited. So they are going to end in a time. So that's the principal side that we have to create the mathematical model. Because if I see the situation, I establish that the sources available inside self-free system is the same of the maximal expression of our protein to see the change color. So we can see different curves of expression at different concentration of protein. That's the side what we have to establish the mathematical model. And we estimate the Michaelis-Menten behavior like literature that we see. So we was trying with that side and our side, and we have a linear model that can estimate that two behaviors.
And I would like to add that it was a very long process that took us like 2 months in order to get the dynamical models, because we first started with the biochemical reactions, and then we had to transform it into differential equations. And the mathematical complexity and computational power it took to solve that, all the equations we had, was too much. So we had to make some assumptions, like making some reactions, consider them too fast to not be even considered in the model. We have to make some reductions, invariant reductions, and all those things in order to decrease the complexity of the model.
Thank you.
Thank you. Any more questions from the judges? Yes, Carol, go ahead.
I saw that your project use optimization to try to improve your parameters. This process for you was easy or was really hard to work with the optimization process?
Yes. Fortunately, we had a lot of time, and our advisors also helped us with tips and some classes. So yes, it was difficult, I have to say, but it was worth it. I would like to show how these digital twins software applications we developed worked. So please, can you allow me to share? So basically, we developed these digital twins, incorporating laboratory data in our dynamical models. Why we decided to do that? Because they will help us, these digital twins help us to characterize the quorum sensing and improve the results we are going to obtain from the self-free extract. So here we have the 2 experimental conditions that we have in 2 different, it is a 20 microliter bioreactor and 2 different media, LB and M9. So basically, we have automatization of everything. So here we can let the digital twin calculate the optimal time of harvesting in order to obtain the maximal amount of ribosome by clicking on. And we also know that we can have different concentration of polymerase and PUR according to our needs. So for that reason, we included a library of different RBAs available for Nigen, and we can change them. In this case, for our digital twin, we have to place the dot line here on the dash line by changing the RBAs. This will change the concentration of the protein produced. In this case, we decreased, and on the other case, we have to increase the RBAs for the production. And this will help us with the self-reaction. But not only that, we obtain these conditions, but we also develop a digital twin to characterize our putrescine biosensor label. In this case, we have to input different conditions such as polymerase, ribosome, the transcription factor PUR, amino acids, nucleotides, and the biosensor DNA. So with this digital twin, we can change different conditions, such as the concentration of putrescine, which is written in millimolar. For example, let's suppose a case of 6 millimolars of putrescine. And now we have here 4 different charts with 2 colors of lines. For the blue line is for the circuit that senses low putrescine levels, and the red line is for the circuit that senses high putrescine levels. So in this case, 6 millimolar is a low concentration of putrescine, and we can see that the reported protein is being produced highly, while the red chromoprotein is produced low. Also, we designed, or we have what we expect to see in the label, which is the image we see here, in which case only the blue color can be seen. But as we increase the concentration of putrescine to a toxic level, let's say 24 millimolar or even 33, we see that both production of chromoproteins are high, the blue and the red one. And in this case, this will tell us that the food is not safe for consume. So this optimization is what the digital twin helps us. And this is also helpful because we don't have to go to the lab and try different experimental conditions and waste a lot of resources. Well, we can do it with the dynamical models and experimental data.
Thank you. We have another question from Kevin, please.
Yeah. So maybe this is a tricky question, but I don't know if different types of meat, for example, chicken or, beef or etc., have different, levels of putrescine. I don't know if that's the case or not. And if you recognize that is the case, how could you solve that in your modeling?
Well, actually, we consider, or we initially focus on beef. Why? Well, happens, yes, chicken could be a good option because it's a lot of consumption in Ecuador. However, we focus in beef because this kind of meat has better nutritional conditions. So for now, we focus in beef. However, in the future, we will implement for different products like chicken, like pork, or even sausages, because the principle will be the same. However, we must make some adaptations and changes in the parameters. Just only with the digital twins, we can adapt for different purposes, and we can try in the laboratory stage in wet lab in the future.
Hey, thank you.
Thank you. Do you have any more questions from the judges? Yes, Harold, go ahead.
Your genetic circuit detects a high quantity of putrescine molecules. What happens if or do you have any scale to detect the quantity of putrescine, or only detect high quantities.
Yes, of course. For that, we based on parameters that we found on papers. We noticed that there is a different concentration in different countries where putrescine is bad for humans, is toxic is well to say. In our case, we are using the parameter of 19 millimolar, that this concentration of putrescine is toxic for intestinal cell humans. So focusing on this parameter, we design our circuit design and our model. So of course, we know that there are other values, for example, but that's why we're using digital twins for change just the model and change this value.
Thank you.
Thank you. Do we have any more questions from the judges? Yes, Kevin, go ahead.
Yeah, just one last question, and also a really important comment, because I think your project is really complete. It has a lot of depth in it, and I think you should know it, and you should be proud of that. I want just to ask about the final product. If I understand correctly, this product is from only one use, right? You just use it once, and that's it, or is it another like kind of use for it?
So it's 1 year. So basically what you have is the smart label, and due that it's lyophilizated, so that's basically what gives you a longer range of time that you can use whenever you want. However, once you start using it, it will only get the amount of the beef you're using, and after that you'll have to use another smart label.
Okay, thank you.
Thank you. Do we have any more questions? Yes, Bella, go ahead.
Well, felicitations as well on this project. Me too. I find it pretty practical, since you have a product to sell at the end. You don't depend on so much other things. What is your guess as to who the customer might be for this smart label? Would it be the supermarket itself? Would it be the consumer? Would it be an inspector of sanitary services? What do you think about that?
Okay, thanks for your question. We developed a model, a business model, B2B model, business to business model, because we did surveys, and in these surveys, we found that meat distributors distrust in expiration date. They think that they are ambiguity in this expiration date. Also, the surveys tell us that they are willingness to pay more, and they need to enhance their security food.
I would like to also add that we work for this project to submit distributors such as Pronaca, one of the main distributors of meat in Ecuador. And they've shown also interest in how the label could work as an efficient way to analyze the spoilation meat. And also we found some trust in these vendors in relation to our cost-efficient product.
Yes, and finally, we had different meetings with Pronaca, and they told us that they would like to further have interactions with us in order for them to actually supply with people in order to help us build this project. Once this competition has ended, it has a potential to actually start increasing in the market here in Ecuador.
Thank you. We have time for one more question. Yes, Harold.
This is respect with the business model. Do you think that it's possible to change the business to business model to a business to custom model, or it's not possible?
Yes, it's possible, but we can start with meat distributors because we can do that people accept first the product through these companies. And then we can create a business to consumer because the people can understand how it works.
Thank you.
We have one more question from David.
Hi everyone. Sorry, I know very well, but I have a question about this business model. What what would be your strategy for entering the market based on the B2B business model? Probably you think about a patent, for example, or what is your strategy?
We can plan, we have planned first create a conversation with ARSA that regulates the sector of food in Ecuador. And then we can also create a alliance with Pronaca because we offer some advantage for their company, and with this we can enter to the market. So with the first step is develop this in laboratory, then create alliance with ARSA, and then we can sell our product to Pronac.
Thank you. Any last questions or comments from the judges? Yes, there are.
So how long do you have to wait for the colors to appear on the label?
Jesus, can you allow me to share the screen, please? Well, that depends on how you set the label on this in this digital twin. In this case, we only have an approximation of about 30 to 1 hour, but this data comes from, most of the data comes from also literature. So in this case, we have to go to the lab and apply the DBTL cycle of engineering. We have to test different putrescine concentrations and see how our circuit reacts to that. And then we have to make some improvements to the digital twin now with all the variables from the laboratory, and then we'll get a exact and accurate response. But from the digital twin, we estimate to be 50 to 30 minutes to 1 hour, like for here, to be visible.
Also, it's important to note that the label is the focus of the label is to see the color as the meat gets more and more spoiled. Therefore, the color will increasingly appear over time, which is a way to show the label that has a great expense of time to analyze and control the quality of meat.
Okay, thank you. So if we don't have more questions or comments, yes, we have one from Michelle. Go.
Hi everyone, congratulations for your work. You said that your project is cheaper than other methods, but is cost effective?
Well, I would like to start it saying This is cost effective because, for example, until now, we don't have a similar product in the market. So if we compare with analytic methods like chromatography to detect putrescine in the industry, that is so expensive. However, here with this label, we will have the opportunity to detect the putrescine levels with that efficiency in real time and in a really cheaper way. And another thing, what is cost effective? Because we are using a quorum sensing system to produce our cell-free machinery. Because in traditional ways, we need to add IPTG inducer to obtain the production of the interest products. In our case, we will use an automatic production when we have a high density of cells. In that way, we can reduce even the consumption of carbon sources or carbon sources and nutrients in the production of our sulfur system. So I hope this information could answer your question, but it's a little tricky to identify another device to compare directly. But we analyze in different contexts, and for that reason, we consider and we are sure our product is cost effective.
I would like to add something. For example, in the process to contribute our plasmid, we are using Golden Gate assembly, and for that we need to use only few enzymes. So in that way, we also say that it's cost effective because we don't need a lot of steps to purify or transcriptional unit that is going to be used for the label. Furthermore, as we are using a sulfur system that we are producing, we don't need to buy the ribosomes. Ribosomes, remember, that are so expensive if you buy in the merch. So as we are producing that and taking from a cell extract, we don't need to add and we don't have to pay these cost effective, I will say. So we will, furthermore, I would like to say something else. We are using the cellulose for our membranes for the label. So in that way, we are also being ecological and economic system. So we are really happy of our project because we are taking in consideration a lot of parameters, not only in the materials, even though also in the process to create all biosensor.
Okay, thank you.
We are finishing the session now. So I will instruct all of the students from IGEM Biotech EC to please leave the room, and also the advisors, so we can allow the next team to join. Thank you.
Thank you so much. See you.
Thank you.
Thank you
so much.
Thank you.
Bye.
Thank you.
Thank you. So now I will let the students from the next team join the session. Welcome, students from IGEM CSB. If you have any more team members that are in the waiting room, please instruct them to add your team name to their Zoom name so we can let them in. I will start by sharing your promotional video, and after that you will have 20 minutes to answer questions from the judges. So we will start with your video.
¿Sabías que el Perú produce más del 75% de las 4.000 variedades de papa que existen? Sin embargo, los agricultores enfrentan un gran desafío: el tizón tardío. El tizón tardío, o rancha, es una enfermedad causada por un pseudohongo. Este pequeño microorganismo ocasiona muchos problemas en un gran número de regiones peruanas, destruyendo cosechas enteras y afectando gravemente la calidad de los cultivos. Con el afán de controlar esta amenaza, los agricultores peruanos han intentado frenar su avance utilizando principalmente fungicidas. No obstante, esta solución no siempre es efectiva, ya que puede ser costosa y dañar el medio ambiente. Se han reportado casos de intoxicación, daños cutáneos y problemas en neonatos de mujeres gestantes que realizan dicha actividad.
Es aquí donde llegamos nosotros. Nuestro proyecto, UIRRA, busca enfrentar al tizón tardío de la papa, enfocándose en los siguientes ejes: detección y ataque. Usaremos moléculas fluorescentes que identificarán la pared celular del pseudohongo y así detectar visualmente los cultivos infectados. Para combatir la rancha, utilizaremos la función de una enzima propia de la papa, que destruirá las uniones de su pared celular. En un futuro cercano, es posible que presenciemos algo increíble gracias a la biología sintética: embriones de papa protegidos del tizón tardío. Esto sería un salvavidas para los agricultores, ya que no solo favorecería su salud, sus ingresos y la calidad de alimentos, sino también ayudaría a reducir el impacto ambiental de la agricultura. ¿Te imaginas lo emocionante que sería? Únete a nosotros en la lucha contra el tizón tardío. Juntos podemos hacer la diferencia y construir un futuro más sostenible. Apoya al equipo IAM CSB y ayúdanos a proteger la seguridad económica y alimentaria de los agricultores peruanos.
Thank you. So now we have 20 minutes to reply questions from the judges. We have the first one from Kevin.
Hi everyone. First of all, amazing videos, both really great edited, really amazing. I have a question because you did a great work with the human practices. I want to know how your findings on human practices could contribute to create a method to better deliver your proteins to the final, the terrain, the soil.
We can hear you.
Hi, can you hear us?
Yes.
Good morning, everyone. I would like to answer that question. First of all, it was really good to be part of this project, and human practices got a lot of work to do here. First of all, we traveled around Peru to know what cities were the more affected because of this situation, this disease of potato. So first of all, we traveled to Huánuco, and there we got a lot in touch with the agriculture that got this problem. So, and they told us what they knew about this issue and what they did to address it. And we were really, it was really impactful. So we got a lot of things to do. So human practice got campaigns of information. We got a lot of science communication going on. So in there, we involved not only the agricultures, but we involved also the schools. So the schools got a lot of communication from our team. And there we expressed what we wanted to do and what our solution, our proposal for this solution was. So I think the methods were not only for the agricultures, the people that were more affected, but also for the people that live in the city and they were like really affected because agriculture plays a really big important role in Peruvian economy. So yeah.
Thank you. We have one question from Michelle.
Hi, everyone. Congrats for your work. I am Peruvian, so when I saw your video, I was emotional because this not only affects Peru, this affects the whole world because we all consume potatoes, right? But in the Peruvian context is a little complicated. For example, for the transgenic moratorium, the CRISPR-Cas patent, and other things. My question is related to the previously. Given the public limited knowledge on biotech, synthetic biology, do you have, or how do you plan to educate farmers and community about your project?
Yes, we had a lot of campaigns, as I said, and we got a lot of, not only in Huánuco, but here in Lima as well. And we had, and we know that this problem is not only a Peru problem, it's a worldwide problem. We know history, and it also affects everyone. So we did something research, and we had this science communication on Instagram and other social networks. But I think that first, what really impacted us was to travel to Huánuco and around Peru. There we talked with some farmers, and they told us that there was a little bit of misinformation and the lack of presence of the state. They don't help a lot to the state, to the farmers, so they did some really big issues with their own health. They had a lot of health problems because they used some pesticides that were not really that. They don't have the biosafety measures to achieve that. So we got a lot of work to do right there. So.
And yes, from part of the biosafety, we just not only went to Huánuco, we went to organizations, CIPS, that is a really important organization here in Peru that it studies this problem. My teammates, we had a really conversation with scientists. They give us a really good overview of this problem, and they saw our product, they saw our experiment that we want to develop, and they gave us a really good they gave us a really good insight. They told us that it was a really great project, that it develops the global problem that, as you have seen, we want to solve. Not only we just don't want to solve the problem itself. We want to prevent, we want to attack, and we want to detect. So it's a really innovative solution to this problem.
Thank
so I'd like to add something else. Hi, everybody. Apart from all of that my teammates have mentioned, we also have some support from national institutions. For example, the INIA, which is Agricultural Investigation Research in Agriculture. So we send them our proposal of how was the idea of the project, and they kind of validated that. Okay, they were even eager to help us to make this a reality, to make this able to apply in the communities, in cultivars, etc. Also, we also have a few conversations with CIP, International Potato Center, and they were even eager also to test, for the beginning, to test these products in some in cultures, in crops, in situ crops in the institution. So we have we really had a lot of insight and help from these national institutions. So that was really some support for our project. Yeah.
Okay, thank you.
We have now a question from David.
Hi, everyone. I'm also Peruvian. Specifically, my grandparents, curiously, is Yata, Huánuco. I know the project is very excited for me. I want to know exactly how is your plan to scale up the development. Specifically, can you show us about for example, the indicators such as, for example, internal rate of return or net present value, maybe, is very important to recognize about that. Thank you.
Could you maybe repeat the question? I understand there was a focus on scaling the products for the production, I believe so, right?
Yes, to scale the production is very important to understand indicators to revenue, for example, internal rate of return or net present value. Is it possible to show or not, or at least a budget maybe?
Okay. Yeah, bear with us a moment, please. We are gonna have the specialist in that part in Spanish, and we are gonna translate it on the way. So just bear with us a moment, please.
Hello, lo siento, hola, ¿qué tal? No, mi inglés no es muy bueno, entonces mis compañeros van a tener que traducir mi respuesta. En el área de escalamiento, lo que sí teníamos bien presente que la circulación de un producto en Perú puede llegar a tener un poco de dificultad. ¿Está ahí?
Yes, what my partner said that circulation of the product in our country could be quite difficult.
Bueno, lo que nosotros hemos estado percibiendo para la circulación de nuestro producto, o al menos el escalamiento del producto, es enfocarnos principalmente en la etapa en la que estamos actualmente.
So, as a solution of this problem, we want to focus on what we are doing now, on this stage that we are now.
Porque en donde estamos actualmente es en la validación de nuestro producto. Entonces, según asesores externos que hemos visto, donde hemos estado preguntando, investigando, lo que nos han propuesto es que inicialmente nos estemos enfocando en lo que va a ser la validación del producto. Y es
So we have consulted to external assessors, and they told us that we have to we want to focus. They told us that we should focus on the experimental validation of this project first.
Es por esta razón que donde nos hemos enfocado es en encontrar tanto investigadores que nos apoyen como el investigador del CIP, como además asesorarnos con investigadores externos que nos ayuden a visualizar cómo va a ser nuestro protocolo, nuestro financiamiento inicial para lo que va a ser esta investigación, para luego, con esos datos, poder entonces ir y seguir investigando para la producción y futuro escalamiento del producto.
So we have consulted this kind of assessors, and they told us that we should first, as I said before, have the validation, the experimental validation. So then we can proceed to see our financial foundings and funds.
Solo, bueno, también lo voy a decir en español, pero es importante, o sea, no todo es innovación, ¿sí? Hay que adelantarse también en cuanto a la rentabilidad para sobre todo reconocer si es escalable o no el proyecto, y esto se basa en un flujo de caja proyectado. Lo pueden hacer en base a sus presupuestos, costos fijos, variables, etcétera. Pero bueno, está muy bueno el proyecto. Gracias.
Thank you.
I also
Sorry,
like to add something else.
We'll move on to the next
particularly
question.
with the funding of the project. There are several.
We have
programs
questions
in Peru.
from more judges, so we'll move on to the next question, please. So we'll move on with Harold.
Thank you. I really like your project, especially for the social perspective that it has. My question related to your project is, you use a DVTL cycle to develop the project. What do you think that was the best advantage of using this DBTL cycle in the project?
All right, thanks for the question. First of all, we try to have an holistic approach to the problem. So that's why we designed these three axes: attack, defense, and prevention. So that was a key part of the design. So in order to do that, was a lot of research, was a lot of discussion with the team. So particularly the design was something, a key point in the project, because we needed to analyze what could be the best possible solution for each of the axes. Addressing the attack, for example, degrading the cell wall of Phytophthora infestans, some prevention to attach these molecules, these ENF factors, that could potentiate infection from this pathogen. And also the detection that was quite an innovative part. So we basically designed antibodies, well, single chain fragments, de novo, with a software. So this design part of the DBTL cycle was particularly tedious, and that was a lot of research and work into it. But also the optimization of that, sequence optimization, protein optimization, kind of tests to analyze whether this could link, for example, to certain protein in a good way, in a good manner, and also the expression of these proteins, because for all of these, we design an inducible expression system, particularly the T7 system. So this design and test were hand in hand constantly during the project. So those were, I believe, those were the most key parts of the project.
Thank you. Thank you. We have a question from Kevin.
Yes, thank you. Let's talk about the recombinant proteins. So in the project, you said that this protein for attack, for example, or for recognizing, like they recognize some elements that are kind of common between different fungi, fungi cells. So how do you plan to upgrade this problem in future iterations of the project?
Okay, sure now. We also took a look into that, as particularly in the microenvironment of the potato crops. But also this goes hand in hand with the application of these products. There are several pesticides types, like systemic pesticides, like easy application of aerosol pesticides. So we were addressing this problem by the application system. For example, the systemic fungicides, in particular with the gluconase, the attack axis, could be inside the phloem of the plant, and that could not exude throughout the roots or the shoot. So that way, it could not affect other fungi in the bio microenvironment. Also, particularly detection was like a kind of a special kit, because we were based on an ELISA-like system. We addressed this SCFP fragment to an HRP. This, in reacting with the luciferase, could show a luminescent reaction. So that way we could detect particularly this pesticide. And also, one more thing that is that we try with several proteins, but we choose chitin synthase, but we particularly choose the extramembrane domains, because those were the ones who shown the more variability and the more specificity to Phytophthora infestans. We analyzed that with Lyme, and we took a look into that. So it is specific in particular in the detection, because we only address these extramembrane domains. And also, for the prevention part, this was the more specific in particular, because Phytophthora infestans secretes many elicitins, okay? ENF2, 2A, 2B, 1, 4, I believe. We made a docking with each and every one of them. So this one is specific from pain infestans, and the ELR receptor, elicitin-like receptor, binds specifically. So this prevention system prevents these early stages, because these elicitins are secreted by the pathogen in the early stages of infection. So we address each and every one of that. Maybe in the gluconase, it's more related to the application of the product. No, and that's it.
Thank you.
Thank you. We have 2 minutes. Yes, Beda, you have one question. Go ahead.
So, thank you. Also, congrats on this project, which I find really interesting and worthwhile, and don't get overwhelmed by the complexity. Also, with regard to the application, what would the vehicle be? Would that be a watery spray or a suspension? And did you think about the permanence, the time period? This protein, if I understood correctly, stays on the leaves or on the plant? And is it absorbed into the plant?
Okay, well, yeah, we kind of evaluate different application systems. For example, foliar applications was one of the first approaches we tried. But there was a problem with these pesticides could affect microenvironment. So also we took a look into systemic pesticides, as I mentioned before. So that way this could not affect any other roots aside, for example. And also, particularly with that question, also was important the delivery of this product, as you mentioned. For that sense, our proteins have different biochemical parameters like pI, maybe charges, etc. So in that sense, we try to accommodate these proteins in a medium consisting of osmolytes. Osmolytes are kind of ions that could stabilize these proteins for a long period of time. In that sense, this could be easily transported, easily applicable in different environments, because Peru has a ton of environments, from deep cold to extremely hot, and potato crops grow in each and every one of these environments. So in that sense, these osmolytes and stabilizing the protein was key to our analysis, and also the application system.
Para poder adicionar a esa respuesta, también debemos tener en cuenta que para poder comercializar o hacer llegar a las personas este producto, nosotros también vamos a acompañar ello del lado de la capacitación, de una campaña de capacitación.
Well, my partner said that as this could be really complex, we should give some training session to these farmers, because as we mentioned, this problem is really local, and we should not forget how to instruct very well to our farmers, so this product could be really well used.
Dentro de nuestro contexto también existen ciertas instituciones, por ejemplo el SENASA, que también clasifica estos productos dependiendo de la peligrosidad o de qué tan dificultoso es su uso, ¿no? Entonces, yendo de la mano con esa clasificación y con esas advertencias o sus parámetros y la capacitación, esperamos que, bueno, la aplicación sea la ideal, ¿no?
Well, there are other institutions as SENASA that give some kind of instruction, and they can target these pesticides as they're risky. How risky are these kind of pesticides? So we hope that with this kind of help and with the training session that we should give, this product could be really helpful.
Thank you. We have one last question from Harold, and then we will end the session.
Thank you. In the DBTL model, you have the design part, and you have some different equations to the mathematical model. What were the assumption and consideration that you take into account to develop the mathematical model?
Yeah, sure enough. I'm going to translate what my partner says in that mathematical apartment, right?
Acerca de las consideraciones que se está teniendo, acerca de las consideraciones que se está teniendo en la parte de modelamiento matemático, es que se está utilizando en sí un sistema inducible para regular también la expresión de las proteínas, así como la producción de la polimerasa T7, ya que va a ser una polimerasa de alta expresión. Así.
Okay. So the first part of this analysis for the design of these mathematical models was the expression of the T7 polymerase. This inducible system is particular with the T7 polymerase, T7 promoters in our plasmid that we are going to insert our products, and also some regulation of these polymerases.
¿Cómo se regula esta polimerasa T7 a través de la lisosima? Ya que va a formar una especie de complejo, el cual va a inhibir también la producción, o sea, la unión de la polimerasa al sitio de transcripción.
So the first part of the regulation is that a T7 lysozyme, which is already in the strain, I'd like to address that. These systems, these E. coli, these strains, have already the cassette D3 and also the Pilis plasmid. This Pilis plasmid has the lysozyme, as my partner mentioned. This lysozyme forms a complex. We modulated that complex, the T7 lysozyme and the T7 polymerase, in order to inhibit basal expression of the T7 polymerase, in order to only have inducible expression of our proteins.
Y para seguir con eso, una vez ya se mantenga regulado también la expresión de la polimerasa T7, así para que el sistema tampoco colapse, el cual la polimerasa va ya a seguir con la producción de las demás proteínas 23, ¿no? Como las beta-glucanasas, los anticuerpos y los receptores para poder mantener todas estas producciones en óptimo estado.
In order for the system to not shut down, we also modulated this T7 polymerase, how it was binding to the promoter, T7 promoter, in our PET plasmids, which could express our glucanases, our antibodies, and our ELR receptors, well, the fragment of the receptor.
Y también se está considerando el número de copias del plásmido, ya que dentro de la E. coli únicamente el plásmido que lo estamos utilizando, el PET, va a tener un aproximado de 10 a 20 copias. Por eso se realizó un promedio. Y justamente dentro de las ecuaciones diferenciales se está considerando todos estos parámetros también.
So finally, and one of the most important aspects of our project is that this PET plasmid is a medium-low copy number, 10 to 20 copy number of plasmids in the strain. Also, that is considering the equations as a mean, as an average, and it's also considering the parameters to modulate our expression of our recombinant proteins.
Thank you. Thank you. Now I will ask for the team members for this team to leave the room. Thank you very much for your participation.
Thank you very much.